Possums have a complicated relationship with humans. In Australia, they are protected following years of habitat loss. But in Aotearoa, New Zealand, possums have become a serious pest. Every night they eat through thousands of tonnes of native vegetation and fauna and have damaged forest ecosystems. To combat this pest in New Zealand and safeguard possums in Australia, additional tools are badly needed. In collaboration with researchers worldwide, University of Otago scientists have sequenced the entire genetic code of the brushtail possum, annotated their genes, and uncovered where they are switched on and off.
We now have the blueprint to study possum populations in real depth across Australia and New Zealand. The possum we sequenced was a male pouch young from right here on the Otago Peninsula. He and many other possums from southern New Zealand are actually hybrids with heritage from both Tasmania and mainland Australia. Even though all possums in New Zealand came from just a few hundred individuals, the possums we tested have more genetic diversity than those in Australia because they came from multiple Australian sources and have now hybridised. We found lots of other surprises directly coming from the DNA and how it is expressed. Possums are nocturnal, so non-visual communication is really important. We uncovered possum genes responsible for carrying scent in urine and found that although they are silenced in newborns, they are switched on in adults, particularly males. Molecules produced from these genes could be used to lure possums towards a trap or keep them away from pest-free areas.
But possums are important not just for pest control and conservation. They are a model for how the other mammals, that is, marsupials, undergo reproduction. They give birth to tiny, underdeveloped young that crawl from the birth canal to the pouch using only their forelimbs, before undertaking the rest of development suckling from a teat. In many ways, leaving the pouch is a more significant developmental transition than birth itself. But until now, this transition has been very poorly studied.
We undertook the first genome-wide analysis of gene expression changes associated with the milk to herbage dietary transition in marsupials. We noticed a clear loss in the expression of genes responsible for milk digestion. Conversely, genes involved in processing plant-based metabolites dramatically increased. Growth-related genes also changed, particularly those characterised by parent-specific gene expression, a phenomenon known as genomic imprinting. We found imprinted genes that have not been observed anywhere else, indicating marsupials have a rich and complex system to exploit maternal resources that is more focused on post-natal life rather than in the womb.
In the future, we'll use this genome sequence to further understand how reproduction occurs in possums and other marsupials, with a view to controlling it. Possums give birth to young the equivalent of an 8-week human embryo. That's the time at which the reproductive organs are developing. We think this could be the ideal window to access and purify cells of the next generation, something we achieved in this work. In the future, this knowledge could be used to help control reproduction in possums and other marsupials.